Hello, my name is Joshua Kupfer, and today I'll be sharing my ongoing work in the Holloway lab at ASU, calcium phosphate deposition on electrospun fibers as a function of simulated body fluid formulation. Currently, there are a few treatment options for bone tissue defects with two primary choices. The first of these is an allograft. An allograft is a bone tissue transfer from a donor to the patient and carries a risk of infection and disease transfer, while also having reduced osteoinductive properties. The second option is an autograft, where bone tissue is transferred from a healthy site to the damaged site within the same patient. Autografts also have associated risks, including increased infection risk and pain due to two surgeries being required, and they have the inability to treat large defect areas. Still, autografts remain the gold standard in bone defect treatments today. What our work uses are electrospun fibers to mimic the fiber structure of bone in order to regenerate the tissue. Electrospinning uses a polymer in solution being extruded through a needle while simultaneously applying a high voltage to the solution. As the solution exits the needle tip, it is drawn to a negatively charged collector, and while the solution flies through the air, the solvent evaporates, while the polymer elongates into a long and thin fiber on the micro to nano scale. Electrospun fibers have been shown to closely mimic the fiber structure of tissues, such as in the collagen fibrils of tendons. These fibers can also direct cell morphology by causing cells to align along the fiber's paths. Hydroxyapatite also plays an important role in bone tissue engineering. Hydroxyapatite is the primary mineral within bones and has a calcium phosphate molar ratio of 1.67. Hydroxyapatite has been shown to improve pre-osteoblast adhesion and proliferation. In the study conducted by Lee et al., a scaffold with a hydroxyapatite gradient was produced. MC3T3 mouse pre-osteoblast cells were added to the scaffold, and areas on the gradient with higher hydroxyapatite present showed improved cell adhesion and proliferation rates when compared to areas along the scaffold with less hydroxyapatite. Our overall goal is to create a hydroxyapatite layer on electrospun fibers. The first step of the process is scaffold creation via electrospinning. This will be followed by processing steps dependent on the scaffold's material, and the last step is incubating the scaffold in simulated body fluid. The materials being used are polycaprolactone, or PCl for short, and norbornene functionalized cellulose acetate, or NORCA for short. Three simulated body fluid formulations were chosen: conventional, revised, and 4x concentrated revised simulated body fluid. Revised simulated body fluid has increased carbonate ion concentrations while decreasing the chloride ion concentration, bringing the overall formulation closer to the mineral content of human blood plasma. The 4x concentrated RSBF only increases the concentrations of calcium and phosphate ions, which will ideally improve calcium phosphate deposition on scaffolds. The trade-off for these higher concentrations is a decreased pH to avoid precipitation of minerals from the solution. PCl is processed with first an activation step where the scaffold is submersed in a 1 molar sodium hydroxide bath for 6 hours. This activation step oxidizes the surface of the scaffold, preparing it to bind to minerals present in the simulated body fluid. Next, the scaffold undergoes an induction step consisting of a 30-second calcium bath followed by a 30-second phosphate bath. This process is repeated 6 times. The induction steps prime the PCl scaffold to favor calcium and phosphate deposition by only exposing it to calcium and phosphate ions. This creates some crystals on the surface of the scaffold, creating nucleation points for further mineralization of the same crystal type. NORCA is synthesized within our lab, beginning with cellulose acetate and 5-norbornene 2-carboxylic acid. The reaction occurs within DMSO as a solvent using a Bach2O DMAP esterification reaction. The resultant material is crashed out of solution into deionized water before being lyophilized and undergoing a series of washes to purify the polymer. This modification to cellulose acetate is necessary to allow the material to cross-link with a calcium-binding peptide, which is required for mineralization. The ideal NMR of Norca looks like this, with 6 distinct peaks from 3.5 to 5.5 ppm, which represent the cellulose acetate backbone. There are also peaks around the 6 ppm mark, which show the ene group within the norbornene. Norca is processed by adding a calcium-binding peptide to the scaffold. This peptide is photoconjugated to the Norca using UV light. Lastly, any unbound peptide is washed off of the scaffold. The peptide binds to minerals present in the SBF through electrostatic attractions between the charged peptide and the charged ions in the solution. The conjugation reaction between the peptide and Norca occurs between the thiol and the cysteine residue of the peptide and the norbornene group located on the Norca, with I2959 as a photoinitiator. This is a representative image of PCl fibers prior to mineralization, and after a 7-day incubation period in each SBF formulation, the scaffolds look like the following. Each formulation results in a layer of minerals on the surface of the scaffolds, while also bridging any gaps within the fibers. CSBF and RSBF show some gaps within the mineral layers, while 4X RSBF is a more consistent layer. 4X RSBF also shows distinct regions of sodium chloride and calcium phosphate mineralization. These are the two types of mineral deposits that occur on all of the scaffold groups, but there is much more clarity between these two types of mineralization in the 4X RSBF samples. This graph shows the comparison of calcium phosphate and calcium sodium ratios within the 3 SBF formulations after the PCl scaffolds are mineralized. Mineralization is quantified using energy dispersive X-ray spectroscopy, or EDS for short. 4X RSBF shows a calcium phosphate ratio most like that within native hydroxyapatite, with RSBF also being similar. CSBF has the greatest preference for calcium deposition versus sodium deposition, though. Within Norca, peptide concentration was found to affect mineral deposition for CSBF. Using alizarin red S staining, we found that the higher concentrations of peptide bound to the scaffold resulted in seemingly darker red coloration of the scaffold. Within a scaffold with no bound peptide, though, no minerals were present. Using absorbance spectroscopy to compare the stains for 4 different peptide concentrations over varying incubation times, a higher incubation time was found to increase mineral deposition. Both 300 micromolar and 250 micromolar concentrations of peptide had similar maximum values at higher incubation times, meaning that there may be a maximum amount of mineralization possible independent of peptide concentration and incubation time. The key takeaways for PCl are that 4X RSBF resulted in calcium phosphate deposition most like native hydroxyapatite. Meanwhile, CSBF had the most preference for calcium deposition versus sodium. In Norca, increasing the concentration of peptide increased mineral deposition, while increasing incubation time also increased mineralization. When no peptide is present, mineralization does not occur, showing that Norca does not naturally mineralize in simulated body fluid. Future experiments will include testing Norca with peptide in the other SBF formulations besides only CSBF. We also plan on testing the effect of induction steps like that for PCl on the Norca scaffolds prior to SBF incubation. The creation of a mineral gradient along scaffolds will also be explored, and cell differentiation and morphology studies will be conducted on the Norca scaffolds. I'd like to thank the other members of the Holloway Lab, the Eyring Materials Center at Arizona State University, and the Arizona Biomedical Research Center for supporting me in this research. I'd also like to thank the audience for listening.